All right, let's dive in. We're tackling antibiotics today.
Oh, interesting.
Yeah, their effects on protein synthesis in bacteria. It's really complex.
It is a constant back and forth.
Right, especially with resistance growing. We're focusing on the book Antibiotics, Challenges, Mechanisms, Opportunities.
By Walsh and Wenswich, a classic.
Exactly. And I know that's familiar territory for you.
Absolutely. It's essential for understanding these drugs.
Okay, so bacteria need to make proteins to survive.
Right, of course. It's fundamental.
And antibiotics can mess that up, often by going after the ribosome.
Right, the protein synthesis hub.
But bacterial genomes have tons of proteins. Where do we even start?
Well, not all proteins are equally important, right? Some are always needed.
Like the essential ones.
Exactly. Others are only produced when there's a specific signal.
So antibiotics can exploit that.
Precisely. Target the right proteins at the right time.
Not just shutting down everything.
Exactly. And that often means hitting the ribosome.
That incredible molecular machine.
It is responsible for making those proteins.
But these tiny molecules can actually bind to it and mess it up.
They do. It's remarkable how they can interfere with such a complex structure.
So break it down for me. We've got the two subunits, the 30S and the 50S.
You got it. Each one with a crucial role in protein synthesis.
Making them both targets.
Absolutely. Different antibiotics have different strategies.
Let's start with the 30S subunit. What kind of tricks can antibiotics pull off there?
Well, tetracyclines are a classic example.
Oh yeah, those have been around forever.
They have, but they're still widely used because they work against a broad range of bacteria.
How do they manage that?
They bind to the 30S subunit and block the delivery of aminoacyl tRNAs.
Those are like the supply trucks.
Yeah, carrying the building blocks for proteins. No building blocks, no protein.
Pretty straightforward.
It is. But of course, we can't talk about antibiotics without talking about resistance.
Right, that's the big issue.
Absolutely. And with tetracyclines, resistance is a growing concern.
So how are the bacteria outsmarting them?
Well, one way is by changing the ribosome itself.
Mutating.
Exactly. They develop mutations in the 16S RNA, which is part of the 30S subunit.
So the tetracyclines can't bind as well.
Right. It's like changing the lock on a door.
Makes sense. What else can they do?
They can also produce efflux pumps.
What are those?
They're proteins that act like pumps, removing the antibiotic from the cell.
So it's like they're kicking the antibiotic out before it can do any damage.
Exactly.
Clever. So they block access and also control the internal environment.
It's a constant arms race.
Okay, so we've got tetracyclines targeting the 30S. What other antibiotics go after that subunit?
Well, aminoglycosides are another major player.
I've heard of those. Aren't they pretty powerful?
They can be, but they work a bit differently than tetracyclines.
How so?
Instead of just blocking delivery, they mess with the ribosome's accuracy.
Accuracy, what do you mean?
The ribosome normally matches the right amino acid to the genetic code, right?
Right, like a perfect translator?
Exactly. Aminoglycosides cause it to make mistakes, incorporating the wrong amino acids.
Oh, so the proteins are messed up.
Exactly. It's like introducing typos into a crucial instruction manual.
And that's why they're often bactericidal.
Right. Those faulty proteins really disrupt essential processes.
Leading to cell death.
Exactly.
But of course, bacteria have figured out ways to resist them too.
They always do.
How do they do that with aminoglycosides?
One common way is through methylation.
Ah, methylation again.
Yep. They modify the ribosome, making it harder for the aminoglycoside to bind.
So same strategy, different target.
Exactly. Bacteria are incredibly adaptable.
It's a bit unnerving, isn't it?
It can be, but it also makes it a fascinating field to study.
Okay, we've explored the 30S subunit and the tactics of tetracyclines and aminoglycosides. What about the other half of the ribosome, the 50S subunit?
The 50S is a whole other world of targets. Lots of vulnerabilities for antibiotics to exploit.
So what's going on over there?
Well, one of the classic examples is the macrolide class, erythromycin being the poster child.
Erythromycin, I've definitely heard of that one.
It's a common antibiotic often used for respiratory infections.
So how does it work?
Erythromycin and its macrolide buddies bind to the 50S subunit and block the exit tunnel.
The exit tunnel.
Yeah, it's the channel where the growing protein chain emerges from the ribosome.
So it's like creating a traffic jam inside the factory.
Exactly. No way out for the protein.
Effective, but I bet bacteria have found ways around that too.
You bet they have. Methylation, again, is a common strategy.
They really love that methylation trick, don't they?
It's effective.
So they methylate the 50S subunit.
Yep. specifically a certain adenine base in the 23S RNA.
And that changes the binding site.
Exactly. The erythromycin can't bind as effectively.
Always adapting, these bacteria.
It's what they do best.
Okay, so macrolides jam the exit tunnel. What other strategies target the 50S?
Some antibiotics go straight for the heart of the operation, the peptidyl transferase center, or PTC for short.
PTC, what's that all about?
It's where the peptide bonds are formed between amino acids, the essence of protein synthesis.
Sounds crucial.
It is. And one antibiotic that targets this center is linezolid.
Linezolid, okay. How does that one work?
It binds to the PTC and prevents the proper positioning of tRNA molecules.
Those tRNA molecules are important, right?
Absolutely. They bring the amino acids to the ribosome.
So linezolid stops the ribosome from adding new amino acids to the protein chain.
Exactly. It's like shutting down the assembly line.
And is linezolid susceptible to resistance?
It is, but not as easily as some other antibiotics.
Why is that?
Well, the PTC is a very important part of the ribosome. It's highly conserved.
Meaning it hasn't changed much over time.
Exactly. So it's harder for bacteria to mutate it without causing problems for themselves.
So it's a more vulnerable target for us.
It is, but we can't get complacent. Bacteria are always evolving.
I hear you. Okay, so linezolid targets the PTC. What other antibiotics go after that site?
Another one is clindamycin. It also binds to the PTC and interferes with the positioning of tRNAs.
Clindamycin, okay. So similar mechanism to linezolids.
Yeah, but they bind in slightly different positions, which can affect their activity.
Interesting. So subtle differences can have big consequences.
Absolutely. It's all about those molecular details.
Okay, so we've got macrolides, linezolid, and clindamycin, all targeting the 50S subunit.
And don't forget about those combination therapies.
Oh, right, like Cindercid.
Exactly. It's a combination of two antibiotics, dalfopristin and quinipristin.
And how does that combo work?
Well, they both bind to the 50S subunit, but they do different things.
Okay, tell me more.
Dalfopristin binds first and changes the shape of the ribosome.
Setting the stage for quinipristin.
Exactly. Quinipristin then binds and further blocks the exit tunnel.
So they work together to really shut down protein synthesis.
Exactly.
And that makes them more effective than either antibiotic alone.
Right. It's a powerful strategy.
But bacteria can still develop resistance to Cindercid, right?
Unfortunately, they can, primarily by altering the binding sites for dalfopristin or quinipristin.
Through mutations in the RNA, I'm guessing.
You got it. They're always finding ways to adapt.
It's a never-ending battle.
It really is.
Okay, so we've covered a lot of ground with these antibiotics that target the ribosome. We've got tetracyclanes and aminoglycosides hitting the 30S subunit, and macrolides, linezolid, clindamycin, and Cindercid tackling the 50S subunit.
Each with its own unique mechanism of action and resistance challenges.
It's amazing how diverse these strategies are.
It is. It speaks to the ingenuity of both nature and science.
All right, so before we move on to some of the lesser-known antibiotics and their mechanisms, let's do a little recap, make sure we're all on the same page.
Sounds good to me.
Okay, first things first. The structure of the ribosome. Can you give us a quick rundown of those two main subunits?
Sure. The bacterial ribosome has two subunits, the 30S and the 50S. The 30S is all about decoding the genetic message, making sure the right tRNA with the right amino acid gets to the ribosome. The 50S is where the action happens, where the peptide bonds are formed to link those amino acids into a protein chain.
Excellent. Now let's test your memory a bit. How about those antibiotics that target the 30S subunit?
All right, I'm ready.
Okay, first up, tetracyclines. How do they disrupt protein synthesis?
Tetracyclines bind to the 30S subunit and block the binding of aminoacyl tRNAs. It's like cutting off the supply of building blocks. No amino acids, no protein.
Perfect, you've got it. Now let's move on to another big player, the aminoglycosides.
Yes, those tricky ones.
They don't just block access, right? They cause errors in the translation process.
Exactly. They make the ribosome misread the genetic code, which leads to the wrong amino acids being incorporated into the protein.
Resulting in faulty proteins.
Right. And that can really mess up the bacteria.
Leading to cell death in many cases.
Exactly.
Okay, we've covered the 30S subunit. Now let's shift our focus to the 50S subunit. What's the first antibiotic that comes to mind when you think of this ribosomal powerhouse?
Erythromycin. It's a classic.
Erythromycin, a macrolide antibiotic. And what's its claim to fame?
It binds to the 50S subunit and blocks the exit tunnel, preventing the growing protein chain from emerging.
That's right. It creates that traffic jam inside the ribosome.
Exactly.
But as we know, bacteria have found ways to resist. What's their main strategy?
Methylation of the 23S rRNA,
Right, that subtle modification that changes the shape of the binding site.
making it harder for the erythromycin to bind.
Always one step ahead, those bacteria. Okay, let's move on to another fascinating antibiotic that targets the 50S subunit. This one is synthetic, meaning it was created in a lab, not found in nature. Any guesses?
Linezolid.
Linezolid, you got it. And what makes this antibiotic so special?
It targets the peptidyl transferase center, or PTC, which is where peptide bonds are formed. It basically stops the ribosome from adding new amino acids to the growing protein chain.
Exactly, it shuts down the assembly line. And what's interesting about linezolid is that resistance is less common compared to other antibiotics.
That's right. The PTC is a very conserved region, making it hard for bacteria to mutate without causing problems for themselves.
So it's a valuable weapon in our arsenal. But of course, we can't get complacent.
Absolutely not. Bacteria are constantly evolving, and we need to stay vigilant.
Okay, before we move on to those combination therapies, is there anything else about linezolid's mechanism that you think is important to highlight?
Well, it's interesting to note that linezolid's binding to the PTC is different from other antibiotics that target this site. It doesn't directly block the substrates, but it changes the shape of the PTC, disrupting its activity.
That's a crucial detail. It shows that even within a single target site, there are multiple ways to disrupt its function.
Exactly. And that's what makes antibiotic research so exciting and challenging.
All right, so we've covered linezolids. What about those combination therapies? Can you remind us how Synarsid works its magic?
Synarsid is a combination of two antibiotics, dalfapristin and quinupristin. They both bind to the 50S subunit, but they have different roles.
Okay, break it down for me.
Dalfapristin binds first and changes the shape of the ribosome, enhancing the binding of quinupristin.
So dalfapristin sets the stage.
Exactly. Then quinupristin binds and blocks the exit tunnel even more effectively.
A 1-2 punch that's hard to beat.
Right, and that's why Synarsid is so effective, especially against certain resistant strains.
But of course, bacteria can still develop resistance.
Unfortunately, yes, mainly by altering the binding sites for those two antibiotics.
Through those pesky mutations, I imagine.
You got it.
All right, so we've covered a lot of ground here with these different antibiotics and their mechanisms of action. It's amazing how these tiny molecules can target such specific sites on the ribosome and disrupt this essential process.
It is remarkable, and it's a testament to the ingenuity of nature, both in creating these antibiotics and in finding ways to resist them.
It's a constant battle, for sure.
Absolutely, and that's what makes it so fascinating to study. Welcome back. We're jumping into antibiotic resistance now.
Yeah, it's a bit of a scary topic, actually.
It is a serious issue, but it's also fascinating from a scientific standpoint.
How so?
Well, it really shows how adaptable bacteria are. They're constantly evolving to survive.
Right, like a constant arms race.
Exactly. So before we dive into specific mechanisms, maybe we should take a step back, look at the big picture.
Okay, good idea. What are the main ways bacteria fight back against antibiotics?
Well, they have a few strategies. They can modify the antibiotic itself.
Like change it somehow.
Yeah, make it inactive.
So it can't do its job.
Exactly. Or they can change their own target sites.
So the antibiotic can't bind.
Right. Or they can reduce the amount of antibiotic that gets inside the cell.
Okay, 3 main strategies. Modify, alter, reduce. Let's break those down. Starting with modifying the antibiotic. How do they do that?
Well, they can produce enzymes that chemically change the antibiotic.
Like breaking it down.
Yeah, sometimes. Or adding chemical groups to it, messing with its structure.
So it can't bind to the ribosome anymore.
Exactly. This is a common way they resist aminoglycosides, for example.
Oh yeah, those aminoglycosides, they cause those errors in protein synthesis, right?
Exactly. And bacteria have enzymes that can inactivate them.
Pretty sneaky.
They're good at it.
Okay, what about altering the target site? We talked about methylation earlier.
Right. Methylation is one way they do it. But they can also mutate the genes that code for ribosomal proteins or even the rRNA itself.
So they're changing the actual structure of the ribosome.
Exactly. Even a small change can prevent the antibiotic from binding.
So it's like they're changing the locks on their doors.
Perfect analogy. And we see this all the time with macrolides and aminoglycosides. Bacteria methylate the rRNA, and the antibiotics can't bind as effectively.
Right. It's like those methyl groups are little shields protecting the ribosome.
Exactly. And they can methylate both the 16S and the 23S rRNA. So they have a lot of options.
It's a bit overwhelming, honestly. Yeah. All these different ways they can resist.
It is complex, but that's what makes it so fascinating to study.
Okay, we've got modification and alteration. What about reducing the concentration of the antibiotic inside the cell?
That's where efflux pumps come in.
Those pumps we talked about earlier.
Yep, they're proteins embedded in the cell membrane.
And they basically pump the antibiotic back out of the cell.
Exactly, before it can do any damage.
So it's like they're controlling their internal environment, keeping the drug levels low.
Exactly. And these efflux pumps can pump out a lot of different antibiotics.
So they can contribute to multi-drug resistance.
They're a major factor, unfortunately.
Okay, so we've got these three main strategies: modification, alteration, and reduction. But how do they specifically apply to the antibiotics that target protein synthesis? We talk about a lot of different drugs.
Well, aminoglycosides are often inactivated by enzymes, like we discussed, and methylation is a common way to resist macrolides, lincosamides, streptogramins, even pleurometallins.
So basically, any antibiotic that binds to the ribosome is susceptible to these mechanisms.
Pretty much. And efflux pumps can contribute to resistance to a wide range of antibiotics as well.
Including those that target protein synthesis.
Yep. Tetracyclines, macrolides, even linezolid.
It seems like they have a defense for everything.
They do. That's why it's so crucial to understand these mechanisms. If we can figure out how they're resisting, we can try to develop new drugs that can overcome those defenses.
It's a constant battle.
It is, but that's what makes it so interesting.
Okay, so we've talked about the general mechanisms, but let's get into some specifics. Can you give us some real-world examples of how these resistance mechanisms play out?
Sure. One of the best studied examples is macrolide resistance. Remember how erythromycin binds to the exit tunnel of the ribosome?
Yeah, blocking the protein chain from coming out.
Right. Well, bacteria can develop mutations in the 23S RNA.
That's part of the ribosome, right?
Yep. And there's a specific adenine base, A2058, that's a hot spot for mutations.
So if they mutate that base,
it changes the shape of the exit tunnel, and erythromycin can't bind as well.
so even a single mutation can make a big difference.
A huge difference. It's amazing how much impact a small change can have.
What other examples are there?
Well, another interesting one is resistance to streptogramin B antibiotics, like quinipristin, which is part of Cinarcid.
Right, Cinarcid, the combo drug.
Yep. Quinipristin binds near the exit tunnel, too. But bacteria can modify a specific lysine residue on a ribosomal protein called L22.
Modify it how?
They often acetylate it.
It's acetylation. That's another one of those chemical modifications, right?
Exactly. And it changes the shape of the binding site, so quinipristin can't bind as well.
So they're tinkering with the ribosome again.
They're always tinkering. It's impressive, really.
It is, in a scary kind of way. What about linezolid? That one targets the PTC, right?
Yep. And resistance to linezolid is starting to emerge, mainly through mutations in the 23S RNA again.
Even though the PTC is highly conserved.
Even though. They're finding ways to change it just enough to make linezolid less effective.
It's a constant arms race, isn't it?
It really is.
Okay, so we've got macrolide resistance, streptogramin resistance, and linezolid resistance, all targeting different parts of the ribosome.
And all using different mechanisms to evade the antibiotics.
It's a lot to keep track of.
It is. But it's important to understand these mechanisms if we want to develop new strategies to fight back.
So what are scientists doing to combat this resistance?
Well, one approach is to develop new antibiotics that work in completely different ways.
So not just tweaking the existing drugs.
Right. We need new mechanisms of action, maybe targeting different parts of the ribosome or even other parts of the protein synthesis pathway.
That makes sense.
What else? Another approach is to modify existing antibiotics.
To make them less susceptible to resistance.
Exactly. For example, scientists are working on new versions of macrolides that can bypass that methylation site on the 23S rRNA.
So making them effective against those resistant strains.
Exactly.
That's pretty cool. What else is on the horizon?
Well, there's also a lot of interest in combination therapies.
Like Cinarcid.
Yeah, exactly. The idea is to use multiple drugs that target different pathways.
So it's harder for bacteria to develop resistance to all of them at once.
Exactly. It's a promising strategy.
It sounds like there's a lot of exciting research happening in this field.
There is. It's a very active area of research.
Okay, before we move on, I want to touch on something that's often overlooked: antibiotic stewardship.
Oh, that's crucial.
It's not just about developing new drugs, it's also about using the ones we have responsibly.
Absolutely. We need to make sure we're not overusing antibiotics or using them for the wrong things.
Right. We need to educate both healthcare providers and patients about the importance of appropriate antibiotic use.
Exactly. It's a collective effort.
Okay, before we move on to our final segment, I want to test your knowledge on one of those key resistance mechanisms we talked about: efflux pumps. Ready for a challenge?
Bring it on.
Okay, how do efflux pumps contribute to antibiotic resistance?
They're proteins in the cell membrane that pump antibiotics out of the cell, so the drug can't reach its target.
Perfect, you've got it.
Hmm.
Now let's move on to our final segment. We're going to put your knowledge to the test with some exam-style questions.
Sounds good. I'm ready to test my understanding.
Welcome back to the deep dive. We've covered so much ground already.
We have. It's amazing how many different ways antibiotics can attack bacteria.
Right. And all those different mechanisms targeting the ribosome, those essential proteins.
It's a complex system, that's for sure.
But of course, bacteria aren't just giving up.
Oh, absolutely not. They're fighting back.
That's where antibiotic resistance comes in.
Exactly. And it's a huge challenge.
We've touched on some of the broad strategies bacteria use.
Right, like modifying the antibiotic, changing their own targets, reducing the drug concentration.
But let's dig a little deeper into those mechanisms, specifically those that affect protein synthesis.
Sounds good to me. Always happy to explore those molecular details.
All right, let's start with enzymatic inactivation. How do bacteria use enzymes to fight back against antibiotics?
Well, one classic example is with aminoglycosides.
Those are the ones that cause the ribosome to misread the genetic code, right?
Exactly, leading to all sorts of errors in protein synthesis.
And bacteria can actually destroy those aminoglycosides.
Yeah. Well, not exactly destroy, but they can modify them.
Modify them how?
They have enzymes that can add chemical groups to the aminoglycoside molecule or break specific bonds.
So it's like they're changing the antibiotic structure.
Exactly.
Right.
And that prevents it from binding to the ribosome.
So the antibiotic is basically useless.
Pretty much. It's a very effective defense mechanism.
It's remarkable how they can do that.
It is. They've evolved some pretty sophisticated strategies.
Okay, so enzymatic inactivation is one way they fight back. What about target site alteration? We talked about methylation earlier.
Right. Methylation's a big one. They add a methyl group to the ribosome.
And that tiny change makes a difference.
A huge difference. It can prevent the antibiotic from binding effectively.
We saw that with macrolides, right?
Exactly. Macrolides, lincosamides, streptogramins, they're all susceptible to methylation.
And it's mainly the RNA that gets methylated.
Usually, yeah.
Yeah.
The 16S rRNA or the 23S rRNA.
So even those tiny changes to the ribosome can have a big impact on antibiotic effectiveness.
Absolutely. It's all about those molecular details.
Okay, methylation is one way to alter the target site. What else can they do?
Well, they can also acetylate ribosomal proteins.
Acetylation, that's another one of those chemical modifications, right?
Yep. They add an acetyl group to a specific amino acid.
And that changes the protein shape.
Subtly, yeah. But enough to affect antibiotic binding.
We saw that with quinupristin, right? One of the components of Sinercid.
Exactly. They acetylate a lysine residue on a protein called L22. And that makes it harder for quinupristin to bind.
So they're constantly tweaking their ribosomes, making them less vulnerable to antibiotics.
They're very adaptable, that's for sure.
Okay, so we've got enzymatic inactivation, target site alteration. What about reducing the antibiotic concentration inside the cell?
Oh, that's where efflux pumps come in.
Those pumps we talked about earlier.
The very same. They're like little bouncers at the cell membrane, pumping antibiotics right back out.
So the antibiotic never even gets a chance to reach its target.
Exactly. It's a very effective way to reduce drug concentration.
And those pumps can pump out a lot of different antibiotics, right?
They can. That's why they contribute to multi-drug resistance, a major problem.
It sounds like bacteria have a defense for everything.
It does seem that way sometimes. But that's why research is so important. We need to understand these mechanisms so we can develop new strategies to overcome them.
So what's being done to fight back against this resistance?
Well, on one hand, we need new antibiotics, ones that work in different ways, targeting new sites.
So not just tweaking the existing drugs.
Right. We need completely new mechanisms of action, maybe targeting different parts of the ribosome, or even other steps in the protein synthesis pathway.
And how do we find those new antibiotics?
It's a challenge. We need to explore new sources, new environments, new organisms, even synthetic compounds.
Like a constant search for new weapons.
It is. And it's a race against time because resistance is spreading faster than we can develop new drugs.
What else can we do besides finding new antibiotics?
Well, we can also try to improve the ones we already have.
Improve them how?
By modifying them, making them less susceptible to those resistance mechanisms.
Can you give me an example?
Sure. Scientists are developing new versions of macrolides that can bypass that methylation site on the 23S RNA.
So they're basically tweaking the macrolide molecule to make it more effective.
Exactly. And that can make them work against resistant strains.
pretty ingenious.
It is. It requires a deep understanding of those molecular interactions.
Okay, so we've got new antibiotics and modified antibiotics. What else is being explored?
Well, combination therapies are also very promising.
Like Sinercid?
Exactly. Using multiple drugs that target different pathways.
So the bacteria have to develop resistance to multiple drugs at once.
Right. It makes it much harder for them.
It sounds like there's a lot of hope for the future.
I think so. There's a lot of exciting research happening and a lot of brilliant minds working on this problem.
Okay, before we wrap up, I want to touch on something really important. Antibiotic stewardship.
Oh, that's crucial. We can't just rely on new drugs.
Exactly. We need to use the antibiotics we have responsibly.
Absolutely. That means educating both healthcare providers and patients, making sure antibiotics are only used when they're really needed.
Right. No more unnecessary prescriptions.
Exactly. And it also means being careful about how we use antibiotics in agriculture and other settings.
It's a complex problem that requires a multifaceted approach.
It does. But it's a problem we can solve if we work together.
Well said. It's been an incredible journey exploring the world of antibiotics and the bacterial ribosome.
I agree. It's a fascinating area of science, full of challenges, but also full of hope.
Thanks for joining us on this deep dive and for sharing your expertise.
It's been my pleasure. Always happy to talk about antibiotics.
And to everyone listening, remember that we all have a role to play in combating antibiotic resistance. Be informed, be responsible, and be an advocate for the wise use of these life-saving drugs. Until next time, stay curious,